Okay, all right, so we're on notes page 4B, and what we're going to do today is talk about transcription and translation. All right, so we've talked about DNA replication. That's where we're making copies of DNA in the S phase and interphase. And what transcription and translation is for is to carry out the whole purpose for DNA. What is the purpose of DNA? What's its job? It contains the instructions for what, making what? Okay, it does contain your genetic code and specifically proteins. All right, it has the instructions for how to make a protein. Okay, well, there is a slight problem. What organelle is your DNA found in?
Cells.
Well, what organelle in cell is found in the nucleus? Okay, all right. If you'll think back to when we talked about the cell, what organelle inside the cell makes the proteins? Where are the proteins assembled?
Again?
Where are proteins assembled? We've got our cell, we have our nucleus, we have our DNA inside of it. Okay, and what did you just say? Ribosomes. Remember, ribs are protein. Ribosomes make protein. Okay, remember that? All right, so our ribosomes, remember, look like the little dots outside, inside the cell in the cytoplasm, but they are outside the nucleus. So our instructions for making the proteins are inside the nucleus. But the place that we actually make the proteins is outside the nucleus, okay? It's out here. So that presents somewhat of a problem. We've got to figure out some way to get our instructions from inside the nucleus to outside the nucleus to the ribosome so that we can make this new DNA, okay? Or take the DNA and make these proteins out of it. All right, so what we're going to do and how we're going to accomplish that is we're going to use RNA. And this is going to go back to the RNA coloring sheet that I gave you guys the other day, and we did the DNA side, but we didn't do the RNA, okay? But I want to give you these two guys, Beadle and Tatum. They both worked at Stanford University. It talks about that in your notes, the very first thing. They worked in Stanford, or at Stanford University in the 1930s. And they are the ones that determined that genes, and what a gene is, it's a specific nucleotide sequence. So your T's, A's, C's, and G's. That a certain section of those would code, it says one gene for one enzyme is what they originally thought. They thought that these sections of your DNA coded for a specific enzyme. Well, since then, we've revised that a little bit because we know that they code for proteins. Enzymes are proteins, but is every protein an enzyme?
No.
No. Okay? We know that enzymes are proteins, but not every protein is classified as an enzyme. So we changed it to one gene codes for one polypeptide. And if you remember when we played the amino acid bingo, that's what we were doing. We were taking codes, little 3 letters, nucleotides, they're called codons. They code for an amino acid. Those amino acids put together then code for a protein. So those nucleotide sequences code for proteins. So the way that we're going to do that, we're going to accomplish this through your RNA. And we're going to do it through a process, 2 processes called transcription and translation. And that's what we're going to talk about, or what your notes talk about in here today. So I want you to get out your coloring sheet that we did the other day where we had the DNA on one side. We have the RNA on the other side. Okay? Now, RNA is a cheap, disposable copy of DNA. We use it to carry messages, to carry the DNA code, but it is not permanent and long-lasting like DNA is. Now, evolutionary theorists say that RNA was made first because it was simpler, and then as things evolved, organisms evolved, you got your DNA, which was a double strand, as opposed to the RNA, which was more, I guess, not as stable and was more disposable. So I want you to look at your RNA. What's the first thing that you notice about how many strands? There's only one. Okay, DNA has 2. This one has 1. All right. What is something that's different about the nitrogen bases?
It will have another set that matches it.
Before the R.
All right, do you have A's? Look at your shapes. Do you have adenines? Yes. Do you have guanines? You don't have thymine. Thymine is missing, okay? Instead of thymine, you have something that is called uracil.
Did you say urine?
Yeah.
Okay, it is called uracil. So I want you on the shape that looks different. It's got like a little a notch cut out of it. That is a U. So I want you to go ahead and label that as a U. All right. So you should have A's, U's, C's, and G's. And the reason that you have U's is because we just said that we've got to be able to leave the nucleus and take that message, that code for how to make a protein to the ribosome, okay, in the cytoplasm. Thymines are too big and can't get out of the nucleus pores. Uracil is smaller, so that is why we have uracil in RNA, because RNA is going to carry our message for us. It's going to be our carrier. In fact, we call it messenger RNA. It's what we call one of the types of RNA that's going to help in this process, okay? So go ahead and fill in, if you've not already, your A, U, C's, and G's, okay? All right, if you've not already done that. Okay, so you've got your A's, U's, C's, and G's all filled in. Okay, what is going to pair up with the G's in RNA? C's. Okay, cheese grits, grilled cheese. All right, it's still the same rule. All right, Chargaff's rule, remember that? Okay, it still applies to RNA, except, and we're talking about in regards to when RNA pairs with DNA. It's going to pair up with DNA because RNA only has one strand, so we're not talking about two strands of RNA. And I'll explain what I mean in a little bit about that. Okay, what would match up with A's then? We don't have T's. U's. Okay, on an RNA strand, U would pair up with the A on our template strand. Okay, so you can remember that. University of Alabama, Auburn University. You can use that to remember that A's and U's go together. Okay, now there's one other thing that's going to be different. Our sugar is different. What kind of sugar did we use in DNA?
Monosaccharide.
Okay, what's the name of it? It's the D in the DNA.
The deoxyribose.
Deoxyribose. Okay, deoxyribose is our sugar in DNA. Ribose is going to be our sugar in RNA. So I want you out beside your strand there to write that your sugar is ribose. You have an R there. Okay, do you see where it has R labeled for you?
Yes, ma'am.
All right, I want you to fill that in as ribose. So you have your nucleotide is going to be made up of a phosphate, a ribose sugar, and then a nitrogen base. Okay? Now the way to remember those three differences in an RNA strand, and I want you to write this on your worksheet out beside your RNA. Okay? You've got to remember RNA, you are single.
You dang right I am.
Okay? RNA, you are single. So RNA reminds us, the U reminds us that what is there? What nitrogen base is there? Uracil. Okay. R reminds us of the sugar, ribose. And then single reminds us that it is one strand instead of two strands. Okay?
Yeah.
All right. So now I do want you to color these in. Okay, we're not going to do them right this second because I want us to continue on. But I do want you to color those in. You'll use the same color for your phosphate. Your sugar, however, is going to be different because it's a different sugar. It's not deoxyribose, it's ribose. A's, G's, C's, all be the same color. U's will be different. Okay, so the things that are different, you're going to use a different color for. All right. In our transcription and translation, I want you to flip to page 3B, okay? We had quite a bit of room there at the bottom of those notes, And I want you to, we're going to write down the process of transcription, translation. Let me find I guess that's the slide I was thinking of. Okay. All right, here's what happens, okay? All right, you've got this DNA strand. Whoops. All right, you've got this DNA strand, okay? I want you to write that, I want you to copy that down. Draw you a line. Put that it's 3' to 5', okay? And then I want you to copy down those codes. So draw that on your paper. Okay, so let me explain what's going to happen. All right, we said that the DNA strand has our code for making what? Proteins. Okay, so we have the code to make proteins, but DNA can't leave the nucleus. It's too big. So messenger RNA, which is just a cheap, disposable form of DNA, it can carry the message out of the nucleus for us. So you are going to have RNA nucleotides that come and match up with the DNA, okay? So it's going to form a bubble very similar to what we had in DNA replication. But this time it's just going to be messenger RNA that comes in temporarily to get that code, and then it leaves the nucleus, and the DNA is going to go back together. Okay, so this is not a permanent change, but we call that transcription. Do you remember when there used to be scribes? What would scribes do? What was their job? Write stuff down. That's exactly what we're doing right now. Is they're just messenger RNA, which is what this little M that stands for, messenger. It is just copying down the code so it can take it out of the nucleus to the ribosome. Okay? So I want you to come underneath here. Notice that it's going to be going 5' to 3', okay, just like our DNA did in replication. All right, your A, we don't have T's in RNA, so we're going to have to make a U pair with that, okay? So put a U underneath your A. This is called transcription. So I want you out to the side to write, just like you wrote DNA strand, I want you to write transcription out beside here and put that it's messenger RNA. And then I want you to fill in what matches there. And you should be able to fill in those letters without even looking at the slide. You know that A, if it's an A on DNA, it's going to pair with what on RNA? U. Okay? All right, and then we've got, if it's a C on DNA, it's going to pair with a, what on RNA? G. Okay. All right, so what we're writing right now is just transcription, messenger RNA, and then we're filling in our codes.
Wait, why is there a T?
There is a T, that's a good question. There is a T in the DNA. Remember, that's a DNA strand. Here's your RNA. That's where you don't have the T's. So the DNA has a T. And RNA has A's. So it'll match that A up with the T from the DNA.
But that doesn't make sense.
But it doesn't have, now RNA does not have thymines themselves. So they have to use uracils when there's an adenine to pair it with. Okay? All right. I'm going to pause the video and we'll pick up there on Monday. So make sure that you guys have read your notes and that you're ready to finish these on Monday.